I'm Yunzhen from Yunnan Kunming. Yeah, I'm an actual member of the APBioNet. So this is a talk, APBioNet talk series hosted by APBioNet. So this APBioNet Talks is an online education program initiated by APBioNet in the middle of October 2020. The program aims to facilitate open access to knowledge sharing of biomedics. This provides live and on-demand research presentations, tutorials, hand-on practicals, and demos across biomedics. I think experts, early career researchers, scientists, and students from different countries and institutions are invited to sharing about their research projects and skills with the audience. It is collaborated with in Boas, UK, Malaysia, and Blues to provide contents for audience from Asia Pacific, Europe, and Africa. The program encourages contributions from communities and institutional partners. Pre-recorded or live videos are published on APBioNet YouTube channel. More information is also available at the website of APBioNet. Today, we are very glad to have Professor Boas Parker from TU, I think, Braunschweig of Germany to give a presentation about, I think the talk is, you already see, application of long-read sequencing in plant genomics. Professor Boas Parker actually got his training at different universities in Germany. He's doing a lot of research in genomics of plant, including a lot of different projects. And now he is a professor at the TU Braunschweig. The group is about plant biotechnology and biomedics. So next, we were very glad to have Professor Parker to give his presentation. Thank you.
Yeah, thank you very much for the kind introduction, and also many thanks for the invitation and giving me the opportunity to present some of our research here. As you already mentioned, my talk is entitled Application of Long-Read Sequencing in Plant Genomics, and I would like to welcome you all to this presentation. So let's start. Long-read sequencing technology market is currently dominated by two technologies. On the one hand, we have nanopore sequencing offered by Oxford Nanopore Technologies, and on the other hand, we have the SMART, the single molecule real-time sequencing offered by PacBio. So these are the two important technologies that will also appear throughout my talk, and therefore I would like to give a brief explanation how both technologies work. So let's start with the nanopore sequencing. We have here a nanopore located within a synthetic membrane, and a DNA strand, a single DNA strand, or also an RNA strand, is pulled through this pore. And as the different nucleotides are passing through this pore, they are blocking in a specific way this pore and are thereby preventing the flux of ion in the opposite direction. So what we can measure in the end is an electric signal that is changing depending on the nucleotides that are specifically occupying this pore and therefore preventing the flux of ion through the pore. So on the right-hand side, you can see the result. Here we have this electric signal measured over time, and then it is possible to convert this specific characteristic signal into sequence information again. Of course, it's not a trivial process, but it's possible to convert electric information into sequence information. And the most important advantage of this technology is probably the fact that this pore is not degenerating quickly. So the read length is mostly determined by the length of the DNA fragments that you can provide. So if you would be able to provide an entire chromosome in one piece, it should be possible to sequence it also in one piece. The other technology that is used for long-read sequencing provided by PacBio is based on a DNA polymerase that is located in at the bottom of a well, a DNA strand is fed to this polymerase, and the polymerase is producing one new DNA strand based on a template. The incorporated nucleotides are marked with fluorescent groups, and as they are integrated, it's possible to detect this fluorescent signal because only in such cases they remain for a sufficiently long time in this well. And the signal can be seen here on the right-hand side. This is a schematic illustration. We get pulses over time, and every time such a pulse appears, we can assume that the nucleotide was integrated. So this technology also has the advantage that the read length is mostly limited by the provided DNA fragment. So throughout my talk, I would like to address several fields of plant genomics, starting with some Arabidopsis genomics, as Arabidopsis thaliana is the most important model organism for plant research. Then continue with crop genomics, so showing you how we can transfer this technology and also investigate the genomes of crops. And I would like to present several tools that we developed throughout the years that are suitable to process genomic data sets, because these huge data sets cannot be inspected manually in an efficient way, so we need automatic solutions for it. And then finally, I would like to talk about some ongoing projects that we are currently working on. As you can see indicated by the small logos on this slide, some of the work was done at the Center for Biotechnology in Bielefeld, where I worked previously, and some of the projects were now continued or started at the TU Braunschweig, where I started about a year ago. So let's dive into the Arabidopsis genomics then. To investigate the genome of any plant species, we always need high molecular weight DNA. And this is our extraction protocol that we apply for all the species, and it works particularly well for Arabidopsis. So in this example, you can see that we are often harvesting young leaves from a plant. 2 to 3 leaves are often enough. They are grinded in liquid nitrogen, and then the resulting powder is transferred into a specific buffer. It's a CTAP buffer. So some of you might be familiar with the CTAP protocol, and this is an upscaled version that is now, well, we are processing it at a 50 ml scale. So this powder is resuspended in the buffer, then incubated at 70 degrees, and then we go through several steps which involve centrifugation and exchange of supernatant or resuspension of the sediment. And this is like most other DNA extractions. In the end, we have a DNA that can be resuspended in a TE buffer or any other buffer. And the important point here is that we add an RNase. So we incubate our extracted DNA with RNase overnight, because DNA and RNA are very similar biochemically. So it's unfortunately not possible to just extract one or the other, but degrading RNA enzymatically is an efficient way to treat this. Then we continue with quality control. So the first step is a simple agarose gel that you can see down here at the bottom. And on this gel, we can assess if there are sufficient amounts of high molecular weight DNA, the bands shown up here. They are usually even above the highest band of your DNA ladder. And we can also see if there is RNA contamination that appears around 500 base pairs to 1 kilo base pair on the gel. So if you see bands in this region, then this is an indication of RNA contamination. So additional RNase treatment is needed. If everything looks fine on the gel, we continue with quality control and quantification using nanodrop and qubit. So nanodrop is really convenient for first assessment to get an idea about the DNA extraction, if it worked or not. And then the qubit is more appropriate for precise quantification to see what is the precise amount that we can use for the next steps. Then there is something called SRE. This is a short read eliminator kit. This kit is a solution containing different salts. And this solution removes all DNA fragments shorter than 10 kb and depletes fragments shorter than 20 kb. So in the end, we have a DNA sample which is enriched in fragments longer than 20 kb. And we are looking for such large fragments because if we provide large fragments to the sequencing, we also get long reads, and they are usually desired. Well, and then the sample is ready for library preparation and sequencing using one of the long read technologies. This protocol is also available on protocols.io. So if you are interested to apply it in your own research, you're welcome to do so. So then we dive into the first Arabidopsis genomics project, sequencing the Arabidopsis thaliana accession ND1, which stands for Niederzenz1. So most of you probably know Arabidopsis thaliana, but when you think about it, it's usually Columbia 0, the reference accession that you know. And we sequenced a different accession to actually get some insights into the diversity within the species. And here you can see the results of our APBio sequencing. So the long reads, the read length distribution is shown here. This was done a few years ago. So today you would see an improved read length distribution with even longer reads. But even back at the time, we had read lengths up to 70 kb. So that's quite a lot and was sufficient to produce de novo genome sequence assemblies for ND1. And this works by basically extracting multiple copies of the genome, then you sequence it in different fragments as shown here, and you get reads. These reads are overlapping and can be used to reconstruct contigs, which are continuous sequences representing a fraction of the genome. And of course, we need to assess this. So what we usually do in genomics projects, we run multiple assemblies using different assembly tools, in this case, Canu, Falcon, Miniasm, and Fly, and then we compare the results. And there is no unique way how to determine which assembly is actually the best one. It's possible that you look for the number of contigs. You can also look at the assembly size, or you can look at various length distribution metrics. In this case, the assembly size was very important for us, so we decided to use the Canu assembly as a representative assembly. It has the largest assembly size, so 123.5 million base pairs of the Niederzenz1 genome are represented here. And this exceeds both the reference sequence of Columbia 0 and also the results of all the other assemblies. And then we have some length metrics which can be used to assess the assembly quality. I don't know if all of you are familiar with this. This is why I'm including some illustrations about it here. So an assembly can be assessed by sorting all the resulting contigs by decreasing length, as it's shown here. And then we can identify one particular contig where all the longer contigs and this one account for 50% of the total assembly size. And this is called the N50. And then we can look at the length of this N50 contig, and that's given here. So we can say some kind of an average contig has a length of over 13 million base pairs. And 30 million base pairs in the context of a 30 million base pair chromosome already indicates that we assemble entire chromosome arms into a single contig. So of course, there are some challenging regions remaining, like the centromeric regions or nucleus organizing regions, which are not fully resolved, but most other parts of the ND1 genome are represented by these contigs. Then, of course, we can use our assembly to perform meaningful comparison against Col0. So we are interested in differences. How much diversity is there in the species? Are there any large rearrangements and so on? So here you can see a comparison of our ND1 genome sequence on the x-axis and the Columbia 0 sequence on the y-axis. In this case, we are looking at chromosome 4 and just the north of chromosome 4. So when we now would like to compare two genome sequences, it's performed as indicated in this illustration here on the right-hand side. So we basically take small fractions of one of the pseudochromosomes, so of our sequences, and we compare it against the other one. And then we can see if they are largely syntenic, so if the order of all the elements in the genome is the same in both of them, we end up with a line that looks like this. So we have dots forming a diagonal line. And this is what we also see here in our analysis. There are some dots following this line. In this case, it's also combined with some kind of a heat map where a dark blue color indicates high similarity and a lighter color indicates lower similarity. So if there are light blue or even white dots, then this indicates the similarity in this particular block is very small. Right. And then, of course, we can have a situation where the line is not looking like this, where they are not completely collinear, but where some elements are inverted. So this means there is a difference between the one chromosome and the other chromosome. So they are not collinear in this region, but instead we see an inversion. And again, this is what we see here. We have about 1 million base pairs of sequence inverted between the reference accession Columbia 0 and our accession ND1. So then we compared this, of course, against previous reports in the literature, and another study found a similar inversion between Columbia 0 and another accession, which is called Landsberg erecta, or short LAIR. And they identified that LAIR and Col differ by a 1.2 million base pair inversion that's shown here. And of course, this is different to the inversion that we identified in ND1. And when looking in the population, we were able to identify that there are indeed two different alleles that are spreading in the Arabidopsis thaliana population. So there is this 1.2 million base pair inversion that was discovered in LAIR, and the 1 million base pair inversion that we found in ND1. So there is some diversity within Arabidopsis thaliana within a species when we look at different accessions. And then we decided to look at something more artificial, not a natural accession, but a cell culture, in particular 87. So 87 is suitable to study the evolution of a genome over 25 years because this culture was established more than 25 years ago. You can see what it looks like here in the middle in the picture. It's a suspension cell culture. Cells are grown in the dark in a shaker for over 25 years now. They are growing based on a sugar solution that's feeding them, and they are not doing any photosynthesis at all because there's no light. And we extracted DNA from this and performed sequencing using a MinION and a device that's not so well known anymore, the MinIT. That was a device to perform the corresponding computational analysis, the initial analysis, and also data storage. And this is a setup with which we investigated the 87 genome. And what we discovered were segmental duplications. So here you can see the result of a coverage analysis based on the reference sequence that's available for Columbia 0, and 87 is derived from Columbia 0. It's actually just a few generations following the Columbia 0 sample that was used for the genome sequence at the time. So the cell culture and the reference sequence should be very similar, except for the 25 years of evolution in a suspension cell culture. And here you can see what happens to a plant genome during 25 years under such conditions. We were expecting to find large deletions, maybe the loss of genes associated with photosynthesis and so on. But actually, more or less the opposite is happening. So we see that there are many duplications, large regions that are amplified to a higher degree, which are present with multiple copies. And we quantified this in detail because here you can only see how it's distributed over the genomes. But when we look at the actual coverage values that appear frequently, we can see a region that is present with a single copy has about 50x coverage. This means that 50 reads are spanning one particular position that is only present with one copy. The normal situation in Arabidopsis thaliana is a diploid state. So this means there are two copies, two different alleles, and this would result in about 100x of coverage. And as you can see, these are both not dominant peaks. So most of the regions have at least a coverage of 150x, which would mean 3 copies, triploid, or they are even tetraploid, pentaploid, and so on. So most of the genome are amplified to a higher degree. So some kind of polyploidization is going on in this culture. And then we looked at high impact sequence variants to see if genes are disrupted. And yes, there are some high impact variants that disrupt genes, but since there are so many copies of all the genes, these variants only affect a few number of copies, usually just one copy. You can see here how much support there is for the variant allele and for the reference allele. So this shows that there are usually 3 to 4 intact copies of a gene compared to just one broken copy. So this indicates that after this amplification, after duplications of all the genes, they acquire some mutations that break down individual copies. But overall, we did not find any evidence for systematic loss of genes. Now coming to the next Arabidopsis genomics project, sequencing Gabi cut lines. I assume that some of you came across Gabi cut lines or other tDNA insertion lines before. So when we would like to study gene functions in plants, the general approach was to break down the gene and to look at the mutant. You can see it here on the left. The gene is broken, and then we get a different phenotype. We compare the phenotype, and for example, notice that the plant with the mutation is not growing as tall as the wild type. And then we conclude, well, maybe this gene is involved in some growth regulation processes. And to do this at a systematic level, we need an efficient approach to break down genes in plants. Unfortunately, this was not possible in a targeted way for most of the plant science times in the past. So today you could address it with CRISPR-Cas probably, but previously tDNA insertions were used. And tDNA insertions are inspired by a natural system, the agrobacterium that is transferring DNA into a plant cell. This tDNA is then inserted into the nucleus, into the plant genome. And naturally, this bacterium is thereby hijacking the plant cell to produce and secrete amino acids to feed the bacterial population outside. But this was engineered so that the tDNA can be used as a selection marker and that it's possible to identify many plant lines where tDNA is inserted into a particular gene. And this method was used for decades and generated in total over 700,000 different tDNA insertion lines. So we thought it would be really helpful to start characterizing some of them in a systematic way, in particular lines where other characterization approaches failed. And so we picked 14 lines where previous characterization approaches were not successful and sequenced them. We performed sequencing on a gridiron shown here, and you can see one example. So it's really possible to get nice long reads from Arabidopsis thaliana samples with an N50 read length of over 51 kb. So this means that most reads are several dozens of kb long. And in most cases, it was sufficient to sequence just a single sample. Only in one case, we went for a second sequencing run. So this is producing a really huge amount of data, and usually it should be enough to have about 30x coverage, but we got sometimes twice or even more coverage per sample. And then the next challenge was to perform the analysis of these huge data sets, and we developed a tool for it and screened all these lines for tDNA insertions. So the position of the tDNA in the plant genome to find out which genes might be affected. So the idea is that tDNAs are inserted in a gene and that the gene is then broken. And when you have a collection of all these tDNA lines, you can pick the one line that is showing a deletion of your gene of interest. And previous approaches used flanking sequence tags to identify which genes are affected by the tDNA. And we now repeated this analysis using long-read sequencing and identified additional insertions that you can see here on the right-hand side in the table. So it was previously known that about 50% of all the Gabi cut lines display a single tDNA insertion. So on the other hand, the expectation was that 50% of the lines would have more than one insertion. And this is what you can see here. So there were some previously identified and confirmed insertions in these lines, but our sequencing approach revealed some additional insertions shown here on the right-hand side. So the previous assumption was that there are about 1.2 insertions per line on average. But since we now see that we were able to identify 11 previously unknown tDNA loci in just 14 lines, it's likely that this number is an underestimation and that there are more tDNA insertions that were not discovered previously. It's not just about finding the position of tDNAs, but the tDNA insertions can also have effects which go beyond the knockout of an individual gene. For example, here we have chromosomal alterations. tDNAs are inserted in particular positions and they can cause the exchange of chromosome arms. So here we have an exchange between chromosome 3 and chromosome 5, where the small part of chromosome 3 is transferred to chromosome 5 and vice versa. The one chromosome arm part of chromosome 5 is transferred to chromosome 3. So this was observed in several cases. So our 14 lines were not a random selection. They are probably enriched for such more complicated and drastic effects, but we noticed this in several lines. And this can alter the chromosome sizes. And of course, translocations can have many effects on gene expression and thereby also the phenotype. However, we also identified that such chromosome fusion events can take place without a tDNA connecting the two segments. This was previously not identified. So we have, for example, here a connection of a small fragment of chromosome 5 with chromosome 3. And the reciprocal event is connected by a tDNA. So it seems that this is a compensatory mechanism. So if a tDNA is causing the exchange of chromosome arms, then maybe the reverse or the reciprocal event can compensate so that there are no major changes in gene dose. We notice that this can happen across chromosomes, as we see it here, but also intrachromosomal exchange is possible, as we see it here, where south and north of this chromosome are swapped based on a tDNA insertion. So we can conclude from this, although there are large structural variants, we do not really see any cases where the gene dose is substantially altered, and this could indicate that that would be lethal, and therefore we will not find such events. One particular challenging line is the example shown here. So this was the only case where we discovered a really large duplication. So we have not observed any other deletions or duplications except for this one, where about 800 kb of sequence are duplicated. This can be identified when looking at the coverage, so the number of reads that are spanning each position. And this on the left-hand side here is the average coverage, and we can see it's slightly elevated in a region of about 800 kb, and then it drops down to the average again. So this is not so easy to see, but we were able to identify first this event in a coverage analysis, and then we looked at the line in more detail and tried to find out how is this possible. And when we look at the chromosome 2, we find there's a tDNA insertion, and next to the tDNA insertion is an inversion of this 800 kb region. So this region is not just duplicated, but also inverted. And we were able to demonstrate that we can find the breakpoints, or the fusion points, I should say, where the inversion is then fused to the rest of the chromosome. It's a bit further down to the south of the chromosome. And then there is another tDNA insertion, which seems to be completely independent of this event. So here we have two different tDNA insertion loci, a duplication and inversion of 800 kb. So in general, the genome of this line is substantially altered. And after performing this analysis, we proposed that genotyping by long-read sequencing could be a feasible approach to characterize more tDNA lines. So the workflow can be completed within a few days. DNA extraction maybe takes about a day. Then you have to incubate overnight, and library preparation and sequencing takes another day. And then the efficient data analysis, for which we established a pipeline, can be completed within another day in the cloud. So it's definitely possible to characterize a tDNA insertion line within a week. And we calculate also the material costs for this. When you do sequencing using ONT flow cells, you can have 4 consecutive runs on a single flow cell. And this makes the costs about $200 to $300. Of course, it's also possible to use some barcoding approaches and to do this at a larger scale. So it should be possible to do this quite effectively, and that could avoid any confounding effects that might be caused by multiple tDNA insertions being present in one line. So now we are moving on to crop genomics projects, starting with sugar beet. Sugar beet is one of the most important crop plants in the Northern Hemisphere, responsible for about 30% of the sugar production. And the big questions are, how is it possible to secure yield and to increase the resilience to climatic change? When the conditions are changing, the yield should be kept constant. And of course, also it's necessary to develop novel strategies against pathogens. And since genetic engineering is not an option in Europe yet, we need to find other ways through breeding how to enhance these crops. And as a basis for this, we performed long-read sequencing on sugar beet to find more details about the genome structure and so on. So the initial genome sequence was released in 2013, 2014, and since then it's serving also as a model system within its larger taxonomic groups, the Caryophyllales. So here you can see the assembly result. Looking at this assembly, we have less than 200 contigs, and the average size is quite substantial. We have the longest contig, almost 30 million base pairs in length. So again, you can see this is about a chromosome arm, and then we can see the N50. So in a way, the middle contig length is more than 10 million base pairs. So that's showing that we have sufficient continuity to look at many arrangements of genes and so on. We can also assess the completeness using BUSCO. This is something I've not mentioned before. BUSCO is a tool that checks for the completeness of an assembly by looking for conserved genes that are expected to be present in all plants. And we can find more than 95% of all these BUSCO genes. So it's again supporting that the completeness of this assembly is high. And since there are many genetic markers available for sugar beet, it's possible to use these genetic markers to actually place contigs to pseudochromosomes, as you can see it here. So these gray blocks, white blocks, gray blocks, white blocks, they are indicating contigs, individual contigs, and they can be arranged into pseudochromosomes, which are then representing an entire chromosome. As I mentioned, sugar beet is serving as a model system for caryophyllales, so for the entire clade. And here we can see why this is interesting. Why it's interesting, so it helps us to investigate the complex pigment evolution that's going on in the caryophyllales. The caryophyllales are characterized by the production of anthocyanins, a pigment present in plants or in plant families, which are named here in blue. And then there are also beta-lanes present in families, which are shown here in magenta. And the exciting thing about this is that anthocyanins and beta-lanes appear to be mutually exclusive. So if a family has plants pigmented by anthocyanins, there are no beta-lanes detectable, and vice versa. When we have a family that is pigmented by beta-lanes, we do not find any anthocyanins in there. It's even more complicated because the beta-lane biosynthesis is not something that replaces the anthocyanin biosynthesis once, but there are at least 4 independent events of beta-lane biosynthesis evolution, as you can see here by the numbers. And also this beta-lane biosynthesis is restricted to the caryophyllales. You do not find beta-lane biosynthesis in any other plant species outside the caryophyllales. So several exciting facts coming together and thereby representing this complex system. And sugar beet being placed right at the middle of this family as a model system that can be then used to inform genomics projects on any of the other species. And then we come to the next crop species, which is Dioscora dumetorum, also called yams. This is important stable crop in Africa. And here you can see Christian. He brought up this project and he visited us, told us about his ambitious idea to improve one particular yam cultivar. This cultivar is suffering from post-harvest hardening. So after harvest, within 24 hours, this crop becomes inedible because the cell wall turns woody and it's just not tasty anymore and not suitable for eating. So the idea is, of course, to identify the genetic basis of this trait and then to enhance this particular cultivar. So he visited us for a research stay over several weeks. We extracted DNA together and performed sequencing on a gridiron shown here. And then we produced the de novo genome sequence assembly to have a genome sequence as basis for all following analysis. And as you can see, now we have a larger number of contigs. So before the species were highly homozygous or even completely haploid. And now we are dealing with a species which is heterozygous. I will tell you more about that in a moment, but you can already see it from the assembly statistics that now we have more contigs. We have an N50 that is a little bit worse, but still over 3 million base pairs is still high continuity. And you can see the assembly size is about close to 500 million base pairs. But again, we can see with our BUSCO analysis that the assembly is quite complete. 92% of all BUSCO genes are discovered in this assembly, so there is not much missing, if at all. And when we are talking about heterozygous species, then we are dealing with 2 or more different haplophases. So we have one genome copy coming from the paternal parent and one coming from the corresponding mother plant. So we have 2 different genome copies and we can have differences between them, of course. They don't have to be identical. And these differences seem to be distributed not randomly, but usually they are clustered in certain regions. So the differences are indicated here with black dots. We have these differences in regions, which can then in the assembly be separated. So the assembly is producing contigs, which are shown down here in gray. So for these regions, we can have one sequence representing the paternal haplophase and one sequence representing the maternal haplophase. And then we have other regions here in the middle where we do not have any differences between paternal and maternal haplophase. So then it's not possible to separate them in the assembly. And as a consequence, we see in the assembly, we only have one sequence representing both haplofaces. Well, and the consequence of this is when we now look at our assembly and we map all our reads back to the assembly, we can assess what is the coverage in each of the genes. And in many genes, we see a coverage around 100x, and in some other genes, we see a coverage around 200x. So we can now take this information and say if we see 100x coverage, so 100 reads spanning an individual position, we are in a region where we have two copies in the assembly, the paternal and the maternal allele. And then we have some other regions where we have 200-fold coverage, and these are the regions where both haplofases collapsed, and therefore we have twice the number of reads aligned to the same sequence. And this is an approach how you can assess an assembly and see which proportion of it is separated into individual haplofases. So these are some genomic projects. And of course, when we produce large data sets, we have to analyze them in an efficient way. This is what we did through the development of several tools and also benchmarking study. I would like to start with the identification of differences between accessions or cultivars based on read mapping and variant calling. So what you see here is our benchmarking study to look at data sets that to look at tools which are suitable to process plant NGS data sets, so short reads. And what we did, we took FASTQ files, in this case, our NeededSense1 sequencing data sets. We used them for mapping and variant calling against the Columbia 0 reference sequence, using various tools for the read mapping and also for the variant calling. And then we got large numbers of different VCF files. These VCF files contain all the identified variants. And then we performed an evaluation of the different results. And for this, we looked at our APBio-based assembly of ND1 to validate all the variants. So we were able to tell which of the variants are actually true variants and which of them are artifacts or false positives. And by doing this, we were able to compare the performance of all these tools. And while all of them perform quite well, we noticed that BWA-MEM and GATK showed a particularly consistent performance across all the data sets. So therefore, we decided to use these tools for all the following projects working on resequencing data sets. When we have such sequence variants, we have to find out what is their function. So why is it relevant to have sequence variants if we don't know what they are actually doing or contributing to the phenotype? And usually these variants are looked at on an individual basis, so one variant at a time, but this can miss some exciting effects. So we developed a neighborhood-aware variant impact predictor, NAVIP, to investigate sequence variants. Here you can see an example of two sequence variants appearing in close proximity. They are just separated by one nucleotide. And if you just have one of the sequence variants, this would lead to a stop codon. If you have the other sequence variant alone, this would lead to an amino acid substitution. But when both sequence variants come together, T and C substitution, we get a different amino acid substitution. So instead of a premature stop codon that you would assume just based on the one substitution of the T to A substitution, we now get an amino acid exchange, which is probably less severe. And when we looked at all the cases where SNPF, the standard tool for predicting variant impacts, identified premature stop codons, we were able to find in 75 cases where we have two nucleotides changed within a codon, that in many cases, it's actually an amino acid substitution and not a premature stop codon. So of course, we have to consider that two variants can modify each other's impact, and therefore we should always look at all variants that are present in the same allele at least. Of course, this can also be transferred to indels. We have the same situation. If there's just one indel, it is causing a frame shift, and all the following amino acids will be different. If there's only the other indel, the same happens. It's also causing a frame shift, and all the following amino acids will be completely different, and we will see a premature stop codon very shortly after this usually. But if both of them come together and they compensate each other, which means that they are in total inserting or deleting a sequence which has a length that's a multiple of 3, we might only see amino acid substitutions for 1 or 2 of them. So this is shifting. The first variant is shifting the reading frame, and the second variant is shifting the reading frame back to the original reading frame. So we only see changes in a small region. And when we look at this at a broader scale, we can actually find that in many cases where we have such potentially compensating indels, which we call C indels, we see they occur in close proximity, so less than 10 base pairs apart. And this makes sense because a large distance between them would allow for the appearance of premature stop codons, which would disrupt the gene product. The next tool we developed was for estimating the genome size of a plant. So we know about the chromosome structure. We usually have some elements like centromeric repeats in the middle, many single copy regions, but also some transposable elements that are present with multiple copies. Most assemblies are not able to capture this completely. So the assemblies look like this. Some of the repeats are missing, the centromere is not fully resolved, and so on. So when we now would like to know what is the true genome size of a plant, we cannot just say we look at the assembly size, but we need to correct this assembly size. And this is the idea behind it. When we now align all the reads to the assembly, we can see that reads pile up in certain regions. So for example, here on this transposable element, we have reads piling up, or here on the centromeric repeat, we have reads piling up. And when we take the number of reads that are present at such a position into account, we can now calculate how much sequence is missing and thereby predict what would be the true genome size. Of course, we need some single copy regions as a reference to find out what is the average coverage in a single copy region. And we use single copy BOSCOs, genes which are present with only one copy, as such a reference. Then we are, of course, also interested in performing functional annotations. And functional annotations can be split into 2 parts. First, looking at biosynthesis pathways, and then later at transcription regulation of these pathways. So first, we developed KIPES, knowledge-based identification of pathway enzymes, to screen complete genome sequences for the presence of enzymes working in a pathway. We established this for the flavonoid biosynthesis, which is well studied. So the approach is illustrated here. The general idea is first we find candidates based on sequence similarity. We use BLAST for it. Then we construct global alignments using MEFT and build phylogenetic trees. A general assumption is that orthologs tend to have the same function. So we could stop here at the trees. We say we identify orthologs and say that our candidates have the same function. However, there is more information available. For most of these enzymes, we know which amino acids are crucial for the function. So we can inspect the alignments, look at these positions, and check if there is the correct amino acid that would be required for a functional enzyme. And this is what we did. So we are able to predict which enzyme candidates are likely to be functional and which enzyme candidates are not likely to be functional. And this can, in a similar way, be transferred to transcription factors. So we developed another approach, which is identifying transcription factors based on sequence similarity in the first step, then constructing phylogenetic trees to get their relationships, and then looking at specific patterns. For example, for the MIP transcription factors, it's well known what the structure of the crucial MIP domain needs to look like, so we can inspect that. We can also look for other motifs which are specific for certain lineages within this gene family, and so on. So then it's possible to transfer functions which are known from other plant species to the novel plant species under investigation. And with this, I'm coming to the last part of my talk. I would like to briefly mention some of the ongoing projects that we are currently working on in the lab. So there is first the sequencing of the Digitalis purpurea genome. You might know this foxglove plant. It's a beautiful ornamental plant species, and the flower pigmentation is mostly influenced by anthocyanins. You can see some purple spots here. There are also plants which are completely purple and many different other varieties. There's also some medicinal relevance of the species because it was previously used to extract compounds for treatments of cardio diseases. And of course, there's a population genetics aspect. We can see this plant is almost white. So there are white and purple plants appearing in the same population, and that could be studied. It's also an important pioneer plant, which is appearing first at new habitats. For example, after a forest is destroyed by harvesting or by fire, then this could be one of the first plants that emerges there. And we investigated by low coverage OLT sequencing. So we are trying how much of a genome sequence can we actually capture by just low coverage sequencing, so about 10-fold coverage. Then we performed an assembly and screened it for gene candidates. And this is what we are working on at the moment. So finding out how many genes can we find when we just sequence with quite low coverage. And at the moment, you can see the assembly quality is quite decent. So we have about 1 giga base pair, which should be close to the estimated genome size. And then we can find already more than 40% of all BUSCO genes. But of course, we are not so much interested in the BUSCO genes, but more in other specific candidate genes. And the investigation of such genes and gene families is ongoing at the moment. Then we also have 2 projects related to aquatic plants. So this Ceratophyllum is the first of them. It is occurring in eutrophic ponds across Europe. And since most of these ponds are turning into eutrophic ponds, it's also good to study this plant. There are many objectives. 2 students are working on it. So first is the phylogenetic classification of this plant to find out what is its relationship to other aquatic plants. And then it also serves as a system to study the evolution of aquatic plant genomes, what is different between them and land plants maybe. And of course, it helps us to investigate the evolution of gene families and also biosynthesis pathways. And then to resolve the question about the picture I presented on the title slide, Victoria cruziana, it's a water lily plant with extremely large leaves. You can see it here. It's assumed that they can carry a weight of 30 to 60 kilogram. And also exciting about them is that the flower is only opening at night. So here you can see a flower opening in the first night, then it's completely white. And when it opens in the second night, it turns reddish, maybe pink. And after the second night, it's closing, and then it's sinking down to the bottom, and seeds are produced. And the biology behind this might be connected to pollination by beetles. So there's a particular beetle species that is probably moving between the flowers in the night, and then plants are enclosing these beetles during the day. And this is a project where also 2 students are investigating the molecular mechanisms behind it using long-read sequencing. So I would like to summarize the content of my talk. I presented you some Arabidopsis genomics projects as an important model system. We studied this, of course. Then we have several crop genomics projects where it's helpful to get the genome sequence as basis for all further breeding approaches to improve the crops. I presented several tools. Some of them might be useful for your own research as well to handle large genomic data sets. And finally, some ongoing projects so that you can see what we are currently working on. And with this, I would just like to point out some future directions of plant genomics that I see. So first, of course, generating chromosome scale assemblies and phasing haplophases, then looking at the diversity within a species and finding variation that occurs there. Then we probably see a shift to long-read transcriptomics because transcriptomes are more dynamic and therefore have the potential to reveal even more exciting insights. And then, of course, RNA and DNA modifications, which come more or less for free. When we do the sequencing, we could also look for such modifications. And then I would like to finally acknowledge the support from many important people that contributed to all these projects. So my group at the TU Braunschweig, Plant Biotechnology and Bioinformatics, shown also here on the picture in the top right. Then the genetics and genomics of plants group at Bielefeld University, where I worked previously with many members there. And then the CBTech sequencing facility and also the bioinformatics research facility and all the other collaborators at other institutions. And I would like to thank you very much again for the opportunity to present our research here. And if you like to look at the presented publications, I have a list of all of them. And I would like to thank you very much for your attention, and I'm looking forward to an exciting discussion.
Yeah, thank you, Professor Parker. I think we have at least one question posted on the chat part. If possible, yeah, please have an answer.
I have one question here. study and modify the copy of a gene that involves on sweet taste on sugar beet, for instance. I think this question is probably about the sugar production, I think. So as far as I know, the sugar production is now close to 20%, and it's not really feasible to improve it any further because a further increase in sugar content would reduce the overall growth of the plant. So in the end, farmers would not get a higher sugar yield. I'm not sure if I'm getting this question correctly. So the sweet taste is, I think this question is probably just about the sugar, but maybe correct me if I'm wrong.
Yeah, Dr. Farmer, I think, Parinto. If you have a further question, please. Yeah.
There's another question. Haplophase varies for each crop. How will we standardize for each species to significantly visualize the long-read sequence variants? Yeah, that's also a very good question. So the initial idea was to sequence just one plant per species, and then we would have a reference genome sequence. But of course, there are limitations to it. As soon as we are interested in all the variants, it's really hard to very hard to represent the whole diversity. And there are ideas to build pangenomics graphs so that you don't have a linear sequence, but it's more like a network where you have then different graphs going in different directions if there are insertions or deletions, and then it's possible to display the complete diversity in just one data set. So there are certainly ideas to go in that direction. Of course, it's a challenge because if you have a really diverse species, then these data sets would become very large. So if you have to represent all the diversity within a species, and this is why there is a lot of research going on to find efficient data structures for this to store it. Orphan crop. What is an orphan crop? So an orphan crop is a crop species, a species which could be used as a crop, but is currently not really used to full potential. And this is usually due to some limitations that prevent this use. So, for example, in the yam example that I presented here, this is the fact that after 24 hours you cannot eat it anymore. So this is not really good for a crop species because you cannot just grow it to eat everything on a single day. So this requires now some work to modify the plant, and it should be possible to break down this process that is turning the cell walls woody, and then it should be possible to use this plant as a crop, because otherwise it's growing very well in that environment. So it's better to use local plant species and not to introduce plant species which are not adapted to the climatic conditions and so on. And this is what is usually meant with orphan crops, that they are not used to the full potential, that they are somewhat overlooked, and that with some additional work it would be possible to turn species into crops which are not currently used as such. I'm not sure what the question means about register this video. Maybe you can comment on that. Yes, it's possible to study the genome evolution between wild and domesticated plants through plant pangenomics with long read. So I know that there are several projects ongoing. I'm also involved in some of them, and that's definitely promising. So the idea is usually when you have a plant species which is used as a crop, then you can look in wild relatives to find interesting genes like resistant genes against particular pathogens. That's always very promising because you could introduce these genes through breeding, so you don't have to do any genetic engineering. This is, at least in Europe, very important because we cannot sell and also not grow genetically engineered plants here, and consumers do not accept it. So therefore, we need to always find solutions how we can introduce the gene through breeding into a plant. And then it makes, of course, sense to have wild relatives of a crop so that they can be crossed, and that the gene from a wild species can be introduced into the high yield crop lines. what will be the best strategy for polyploid plants. So I assume you are planning to sequence a polyploid plant. So in that case, it's a good idea to have a high accuracy of the reads. So there are two current technologies that you can use for it. So APBio is offering hi-fi sequencing with above 99% accuracy. So these reads are really close to perfect, and that's definitely an approach that you can use. The limitation there is that the reads cannot be longer than 25 KB, and often you get substantially shorter reads. So I'm working in a few projects where we have reads with an N50 read length less than 10 KB, but still these are almost perfect reads. So that's quite good for assembling polyploid genomes. The ONT solution for this would be to go for the Q20 plus chemistry. Q20 plus stands for the Phred score that you get. And with this chemistry, you also get reads that are above 99% accurate. And you have to combine this with the right flow cell type. So the R10 flow cell can be very helpful if you're looking for repetitive regions. And by combining these flow cell types and sequencing chemistry, you can get reads with a high raw accuracy. And this is, of course, a very important basis. And then it can be very helpful if you go for polyploid plants, if you have some information about the diploid parent plants or any related plants which are diploid. And this can then help you to get some information. Of course, it depends if your species is allo or autopolyploid. If it's allopolyploid, it can make sense to also sequence the parental plants so that you can separate your reads before you do the assembly. Something like triobinning, that could be something to look into. And otherwise just try to get as accurate reads as possible so that you can separate the different haplophases. And then, of course, there are some approaches where you integrate genetic marker information. If that's feasible for your species, this can also help you. So then you just need to get contigs that are several hundred KBs long, maybe a few million base pairs, and then you can do the rest based on genetic markers that can help you to assign your contigs to pseudochromosomes. So the next question, what's the advantage is the whole genome sequence for certain plant pathogenic fungi for the control of the fungi and the health of the plant and plant microbe interactions? I think, I'm not working on fungal species, so I cannot tell you all the details, but I imagine it's really helpful to have the genome sequences as well, because then you can, of course, look at the genes that are relevant for the plant pathogen interactions. You can find any genes that can be used as targets by the plant. So maybe you can find particular surface proteins that plants could recognize and so on. But unfortunately, I cannot give you any details about that.
Yeah, any other questions? I think 2 more messages.
Yes, there's one question: how to find the resistant genes in certain plants to maintain the plant's health from pathogens. Yeah, so there are different approaches to this. One approach is called mapping by sequencing. So mapping by sequencing would rely on short read sequencing. So of course you need long read sequencing to have a decent reference sequence, but then you need to have a huge number of plants. Some plants are susceptible to the pathogen and some are resistant to the pathogen. You form pools of them, and then you sequence both pools separately. You have one pool that is susceptible to your pathogen and the other one is resistant to your pathogen. And then when you sequence all of them, you can later map the reads, and then you can find systematic differences between the 2 pools. So of course, each plant will be a bit different and you will have diversity within the pools, but there's one constant factor in each of the pools, and this is resistance or susceptibility to the pathogen. And by using such a mapping by sequencing approach, you find the genomic region that is responsible for this difference. And then you have to look for a gene within this region. And there are some well-known resistant genes. You could look for these genes. Another approach would then be to integrate RNA-seq datasets into this. And of course, you can also use another approach, which would be to look for genetic variation. So if you have one crop species and then you sequence several wild relatives and you know which of the wild relatives are also resistant, then if you assume it's the same resistance source for all of them, you can also look for genes that are present in the wild relatives that are resistant, but not present in the other wild relatives which are not resistant, so susceptible. And thereby you could also explore the natural diversity. Yeah, we have a paper about a mapping by sequencing approach in rapeseed. If you're interested in that, just drop me an email, then I can send that to you.
Yeah, okay. If no more question, I have two more questions. Very nice talk. Thank you very much.
Thank you.
Yeah, the first question is, do your results indicate there are translocation events in just Colombia?
Translocation events in Colombia?
Yeah, in just the wild type, I mean.
Well, you always would have to compare it against something. So when we compare Colombia against any other accessions, of course, we find difference and small translocations, yes. When we just look in Colombia, maybe you mean across generations or between?
Yeah, I mean, maybe different batch of the plants, yeah.
Yeah, that's a very important question. So there are quite a few seed batches around that are all labeled col0, right? And they should differ, but we have never investigated them in detail. So what you could do to do this would be to collect Colombia 0 seeds from across the world from different labs and look for differences between them. This is something that we have not done. But I would imagine you find differences. I don't know how large they would be, but when you look deep enough into this, you will definitely find differences, yes, because we can see that the Colombia 0 that was used to also start our 87 cell culture over time, over 25 years in this case, there are so many genomic differences. And of course, if you are looking at Colombia 0, some groups might be growing it with 3 to 4 generations per year. So you can imagine in the last 20, 30 years, there could be quite some differences.
Okay, I see. Yeah, thank you. And the second question is, I see you estimate some cost for this kind of study. So because the plant genome is very small, I would like to ask, what is your estimation to do the similar study for the human samples?
Well, I think that's complicated. If you go for human samples, it would be medical research. So then you probably have to worry about all the certificates that you would need to do that. If it's just about research, I think a single flow cell could be enough to go with really low coverage at a certain region. But then the question is, what are you hoping to find? I mean, you are not looking for like tDNA insertions in humans. If you already know that there should be a particular insertion in a region, then I imagine one flow cell could be enough. So if you have a good run, nanopore sequencing run, then you can get maybe 15 to 13 gigabases out of a run. So if you are successful with such a run, then you would get about 5-fold coverage for a human genome. Could already be enough to find something, but I would not draw the opposite conclusion. Just because you don't find an insertion, you could not conclude that it's not there. So for the opposite conclusion, you would probably need substantially higher coverage. So it's definitely more expensive, and you could multiply it based on the genome size. So for Arabidopsis thaliana, we have about 150 million base pairs, and the human genome is what is it?
3 GB.
20 times larger, right? So you can imagine how much more expensive it is.
Okay. Okay, thank you. Yeah, I see one more message. What is that? Oh, okay. Yeah, probably this he's asking for some kind of tools. Yeah, please.
Any other tools to look comparison of flavonoid biosynthesis pathway among species in a family? Not sure what kind of comparison you would like to do. So you could run KIPES in a first instance to find the genes that are relevant for the core biosynthesis pathway. And then, of course, it's possible that you have something unique going on in your species. And then it's by definition not possible to identify it based on existing knowledge. But when you look for all the biosynthesis pathway genes that are involved in the core steps, you already have the foundation for your analysis, because then you can go from there with co-expression analysis to find the next steps. For example, you could get all the genes responsible for the aglycon biosynthesis, and then you can try to find additional glycosyl transferases, acyl transferases, and other decorating enzymes that could be involved in your species. And if you would like to compare them, you can also look how are the genes different that are part of the core pathway. And you can look for the regulators. So MIP transcription factors are extremely relevant for the regulation. So that's something you can investigate with the MIP annotator. And we are working on similar tools for other transcription factors. So I hope that they will be available shortly. Is this answering your question? Okay.
Okay, I think it's almost 10 minutes past. We really thank you for joining this talk series hosted by APBioNet, and we would like to thank you again for joining this event. Boas, thank you.
Thank you very much for this opportunity.
Yeah. Thank you. Helia, do you have any question?
No, actually, we would like to provide some information to the participants as well. Maybe for Boas, do you have any last any closing for participants? For example, if they want to connect with you more, like your social account, social media account, or your website, to your lab website, perhaps you could put in the chat box.
Yes, of course. So you can, me and my Twitter handle was already on the slides, right? So you could go for that. So you can find me on Twitter, and lab website. Yes, I would have to copy that. Just a moment. But I can paste it in there. Maybe you can start with your information. So here we are with the lab website. Yeah, so of course, if someone has questions, feel free to contact me, especially if you are interested in using any of the tools, or if you have questions, if you're interested in collaborations and so on. My email address is available on the website, so that shouldn't be too hard to find, and I think you can find everything else from there. I hope you enjoyed the talk, of course, and thanks again for the opportunity.
Thank you very much, Prof. Thank you very much, Prof. Zhang. Thank you, everyone.
Yeah, thank you. Thank you. Thank you.